So we have this session about different geochemical cycles in desert, I guess, greenhouse gas, trace gas emissions, etc. And we have shown Schaeffer coming from Tennessee to give a more invited talk, and a few talks from here, two from my group and two others. And all of us working like all of us Israelis who work here in desert, as I told before, we have very different desert from other parts of the world because we have Mediterranean type cold winter, hot summer desert with winter precipitation only. So when we have water for microbes to be active, they have very cold temperature to be active. And when they have good temperature, they don't have water. So they're continuously under stress. So they stress by temperature, by water. And we will start today. Today we're starting with David Yalin from Weizmann, working with Dan Yakir group, but not on trees, right? The only person.
Okay. Try to stand to that. Does this work? Yeah. Okay. So thank you for this opportunity to speak here, and I'll be presenting kind of work still in progress, so I'll be really happy for feedbacks on what I'm showing here. I'll be speaking about soil carbon turnover and how it changes following drought elimination, a drought elimination experiment. And I realized I should explain this term, the dry timberline. So I'm working for the Dan Yakir group, and in Yakir group they coined this term of the dry timberline. Timberline, we always think about it in the Alps as the high point beyond which you don't get forests, and they said this is a point that we don't usually get forests. That's the edge of the forest for the dry limitation. So soil organic carbon is a giant pool of carbon. We know that it's about double the amount of carbon that's stored as compared to the atmosphere. And for this reason, people in the world are really scared about this scenario of this carbon being lost due to climate change and acting as a positive feedback to climate change. And despite it being such a big pool, and despite people speaking about it for years and years, it's still a very poorly constrained carbon pool. And you can see here, this is not new data, this is from 2013, comparing different models. But you can see that while the models for the ocean are quite well constrained and match each other, the models for the land system are highly variable, almost by an order of magnitude difference. And since 2013, according to what I read, there's still this large variability between models. And the paper just published this year says that the source of the uncertainty is to a large extent the lack of understanding of turnover, both in the plants and in the soil systems. And that's what I'm trying to answer with this research, to give data about how carbon turnover is affected by changes in water availability. So I'll give a very short reminder of the carbon cycle, because I know there's a wide audience here from different areas. Organic carbon reaches the soil through litter, through dead roots and root exudates. And this differentiation, all this organic matter reaches the soil, but some of it goes into a pool which we call particulate, and some to a mineral-associated pool. And we differentiate between them because they have very different turnover times. So we speak about the particulate organic matter going through the soil very quickly, as compared to the mineral-associated organic matter, which is retained in the soil sometimes for thousands of years, and very slow turnover. And we should remember that there's also emissions of carbon dioxide from root respiration, and that's part of the fluxes that I'll be showing that I measure. But for this turnover time for the soil, I'll be looking at soil organic carbon stocks and the heterotrophic respiration and how they're both affected by irrigation. And our hypothesis and the background to the study was a publication from our Yakir group that showed that in the Yatir forest, in the dry timberline, normally the turnover time is extremely high as compared to temperate ecosystems. So we know that this system is usually with very high turnover, and we attributed this with very slow turnover, meaning high turnover time. And previously this was attributed to lack of water for microbial degradation, and we'll see now what happens when we get water. So the research site, as I said, is centered in Yatir. You can see that it's on the verge of being an arid land, on the dry edge of the semi-arid lands in Israel. And we took 1,000 square meters of this forest and spread these irrigation pipes through them, drip irrigation. This is what it looks like from above. And since 2017, you can see there's been a lot of irrigation, much more than precipitation. This was originally intended as a study to see the tree response, but we used the opportunity to see what's happening with the soil fluxes. And you can see the result of this irrigation was much wetter soil, especially in the summer periods. This is soil water content at 10, 20, and 30 centimeters. You can see that in the summers, the soil was maintained wet in the irrigation plot. We used these continuous soil chambers to measure soil fluxes. We situated them next to the tree and in open areas between the trees. And now we've got this fantastic and unique data set of soil fluxes for 3 or 4 years now of this irrigation experiment to analyze. So to start with results after all this background, summer time average daily fluxes, this is the hour of the day, these are the measured fluxes. You can see that in the open areas between the trees, in the control forest, there is even negative fluxes indicating take-up of carbon into the soil during the nighttime, while the irrigated plots gave substantial fluxes in the open areas. This same trend of increased fluxes in the irrigated plot also existed under the tree. Just to mention this uptake of carbon during the middle of the summer, we suspect and we have some evidence for it being abiotic carbon uptake, because we're using opaque chambers, so we can't find any other explanation. And also previous carbon-14 data also substantiates this claim. Overall in the summer, you can see that under the trees there's triple the amount of CO2 emissions as compared to the control plots, and in the open area substantially higher carbon release. I also looked at Q10 values, and I saw that under the trees, Q10 values were lower in the irrigated as compared to the control, meaning that there's another limitation rather than temperature close to the trees. In the winter, the differences between treatments were marginal, and I wanted to focus in the context of this session on what happens in the first rain, but we have very noisy data which I still didn't manage to clean. So instead I'll give you an idea of rewetting in the lab that I just did. And what you can see is that the plot that had a history of irrigation and that we took the soil from to the lab had extremely higher respiration as compared to control soil, and as compared to soil from the shrubland, the natural shrubland in that area. Now, I mentioned respiration so far. Now let's look at the carbon stocks. So you can see from my sampling at the end of 5 years of irrigation, you can see that there's a slight trend of increase close to the trees in carbon pools, but not statistically significant, while away from the trees in the open areas, a slight tendency to decrease, but nothing is statistically significant, so you can say no real change in carbon pools. I looked, as I said, also at specifically the mineral-associated organic carbon, the one which is with long turnover, and also here no statistically significant trends. Away from the plants, you could say there is loss of the mineral-associated organic carbon, but nothing statistically significant. I took all this data and I really wanted to give you some bottom line, what happens with the turnover here. So I did the back of the envelope calculation. I still have to seal all the calculations behind this. But what you can see here, when we look at the annual heterotrophic respiration, these big differences that we saw in the open areas and small differences in the carbon stocks meant there was much higher turnover time in the control plots as compared to the irrigated plots. So in open areas, we can see that almost 5 times higher turnover time. Under the trees, the difference are less significant, but also almost double the time in the control plots as compared to irrigated plots. And overall, we see faster turnover following irrigation. So to summarize, irrigation increased summer respiration, mainly the summer respiration, not the winter respiration. It's decreased summer Q10, just an anecdote to take. No significant effect on the stocks. And as I mentioned, this is 5 years from the beginning of the irrigation. Maybe if we gave it more time to the system, the stocks would change in a significant way. Overall, faster turnover time following irrigation. And the next steps that I'm going to look at are the C inputs to see how the litter inputs changed, trying to understand what's happening here, because my assumption is that if the trees are less water starved, that they are investing less in exudates, for example, and I want to examine that. So that's one topic to still examine. And a topic that we'll have another study to look at is really long-term effects. So now we see that the open areas in the irrigation experiment are now growing plants, and obviously in the long term that will have effects on the open areas will not be open areas anymore. So long-term effects are still to be studied. So thank you guys for this event, to the Yakir group for the hard work they did, and my family, and with that I'll conclude. Thank you.
Very much. What did you show the percent of the soil organic carbon is higher under the trees under the irrigation, but the effect was not significant. Do you know anything about the contribution of microbial biomass to soil organic carbon stock? I would assume that under the trees you have more microbial biomass, and this is why you see the difference in the percent of soil organic carbon and not in the mineral associated organic carbon.
That I count microbial biomass as particulate organic matter, you're saying?
I'm saying, like, when you're measuring soil respiration, does that account for microbial biomass as well, or only, like, what does it account for?
So you're asking, so when I count carbon, I count microbes. They also burn in 900 degrees, so I count them, right? So is it just a much higher population? Maybe, but they're getting their carbon from somewhere, from the trees, right? I don't know. I didn't do any analysis to differentiate microbial carbon as compared to other carbon sources. Yeah. So far.
So far, an effect on the carbon stocks. You don't see it yet. But on theoretical grounds, maintaining those respiration rates that you have, when do you expect to see some with no negative?
So what I expect is that there is more input. So the trees are enjoying the water and their production is increasing and the litter input is growing. So maybe it just balances.
Maybe I didn't ask that far.
Okay.
That was a very simple question.
Sorry.
When do you expect to have no negative?
Ah.
At those respiration rates, when do you expect to see them?
Depletion, or I think even the first year or two. Yeah, I think it would be quite an immediate. I mean, when we speak about agricultural systems and irrigation of agricultural fields, immediate, right? A year or two of irrigation, and you see organic carbon being depleted. So yes. So I think those are the timescales we speak about. Yeah. Sure. No, as far as I know, it's fresh water that they get there, not treated wastewater. So 70 ppm? Maximum 70 ppm. It's 70 ppm. Okay. Okay, that's really interesting.
How you divide it in autotrophic and heterotrophic respiration?
For my calculation here, I used very rough estimate. I used the data that we have from the past from Rafat Khubaja, a postdoc that worked for us, who did it based on carbon 12, 13, 14. He did the speciation. But I'm sure it changed following irrigation, and I need to find a way to retroactively understand it, and I'm not sure I have a good solution for that, but I'll be happy for suggestions. Okay.
Okay, so hello everyone again. I'm Ilya Gelfand here. I'm from, my lab is like 20 meters from here. And the talk is a result of work of Edwige Manjet at last 2 years. And the topic is source of post-rewetting nitrous oxide emissions. And let's start from what is post-rewetting, why is important. So basically, Birch in 1958 published this paper, and since then we have this Birch effect, which just means that after rewetting of dry soil, you have huge emission of CO2. This is kind of Birch effect itself, described in 1958, well before I was born. Now, and source of this carbon is still questioned. So we don't know yet, like 50, 60 years later, what is source of this respired carbon, because respiration is very, very fast. So in 2007, Harvest, right? Harvest is not Jarvis, and it all published that emission of carbon micromole increasing very fast after rewetting and then decreasing laterally, slowly toward 12 days. And Placella, Sarah in the Firestone lab from Berkeley, she published in her PhD that probably this Rewetting pulse is regulated by different actinobacteria rapidly working up rapidly, and they have emission. So this very nice study published in PNAS 2012, and few after, and she showed that actinobacteria working very fast after rewetting, and it's what is driving this pulse of carbon. This is background, this is kind of known. So in our system we have desert and we have rewetting once a year, very important for ecosystem functioning. And a few years back we measured naturally occurred rain. This is rain, this increase in carbon in water and soil. This is temperature, and this is peak of nitrous oxide emissions. We measured nitrous oxide, not CO2, because I'm more interested in nitrous oxide. It's very important gas, it's greenhouse gas, it's also nitrogen cycle gas, very important nitrogen cycle gas. And what we saw a few years back, that after first rewetting we have very fast, very sharp peak of nitrous oxide, and then after second rewetting, although it was much larger pools of water and temperature was the same, we had much smaller pools of emissions, and these emissions work very well with paper published by Austin 2004, and Sean was in this paper too. And the idea is following: we have so nitrogen and everyone dormant during hot summer. Now we're adding water. So the dormant ammonia accumulates. It's also, I showed my PhD a few years back, that ammonia accumulates in dry soils, which is common. Now we have, and fluxes of gases very low. Now we have water. Ammonia start to be oxidized to nitrate by ammonia oxidizers, and we have a lot of fluxes and some leaching, and everything start to work very fast. So okay, all good. But I was suspicious. I had suspicions that this pulse is too fast to be bacterial. This because it's like looks like few hours after water you have already emissions in this dry soil and this very cold temperature. Oh, not very cold, but it's so I had suspicions that it's not organic. And so we went to field and did experiments. How we do experiments? We go to field. We have this few boxes on trolley, and this one box measures nitrous oxide, this one measures NOx, and then we have Lycor for CO2, and we have static chambers, very standard way, and this is how it looks like what we're taking to the field. And then we have static chambers. This is type 1 type, and second type is very standard way to measure gas emission from soil. We're closing chamber, increase in concentration, this is flux, and in this case we have a steady state, and this is also flux can be calculated. Again, it's very, very old and proven technology. Our part we have very sensitive sensors based on laser, which can measure 0.2 and minus 13 nitrogen part per billion. Okay, now this is how it looks in field. In reality, this is in field, water, nitrous oxide, each one is closing chamber, opening chamber, closing chamber, opening chamber. So I just want to show that this is true measurement. It's not something that's in my imagination. Okay, now, so we went to field and we did rewetting. We applied, I think, 6 or 7 millimeters of rain. Isaac can check. Maybe 6-7 millimeters of rain. This is before rain, about 7% water, very dry. Then it's water filled pore space, but it's the same. And now we added water. We have jump of water filled pore space, and then gradually drying out for next 3 days. Now, and we measuring during this time. We measuring CO2 emission, NO emission and N2O emission. And this is mass, but all my graphs same. We have CO2 in red, or whatever it is, N2O in orange, whatever, and NO is in yellow, let's say. And then I don't measure between, so I cannot connect, but I connected just to emphasize dynamics. And you cannot see very well, but we started at 0, rewetting, and this pulse is very fast and then going down over 3 days. And now I'm zooming into this 4 hours after rewetting. This is before rewetting, this N2O, CO2, and NO. We rewetted and we measured once water once it's available, start to be able to measure, and we have very huge jump of N2O, you see, from here to here, and then going down within 1 hour. And CO2 reaching peak about 1 hour. So dynamic is very different between CO2 and N2O. CO2 is like 1 hour delay, and N2O is within first 15 minutes. And there's no way, in my understanding at least, that microbes can work so fast in this desert. So we measured nitrogen in soil, so we measure nitrogen, same, we have jump of nitrogen, then going down slowly, and we measured microbial biomass, also jumping and then going up in 24 hours. And this is kind of regular what happens in dry soils when very wet. Microbes start to be active and they start to reproduce slowly a little bit. And you have more biomass, microbial biomass. But so, yeah, this is just blow up to the same point. You see before rewetting, after rewetting. This is lysis, right? Lysis of microbes, basically. So we can explain CO2 emissions based on our understanding till now, lysis, CO2, acetobacteria, whatever else. And this is known, it's not new. But we cannot explain nitrous oxide, because nitrous oxide within 15 minutes is too fast for my taste. So we came back, Zhu Becker published 2015 a review of abiotic production of N2O, and one of them is ammonia oxidation to hydroxylamine, hydroxylamine oxidation with iron to N2O, no microbes involved. Abiotic, and here attention to stimulated by increasing temperature, which is we have, increasing pH. pH of our soil is about 8.4. A lot of iron. You see soil is a little bit reddish, iron oxide, and salinity, our soil saline. So everything fits well. Never was shown in field nor in lab. It was shown in temperate soil. I haven't seen this paper, by the way, but okay, I believe her. Now, so what to do? We decided to use radiation killing soil without killing soil. I mean, without disturbing anything. So we took this core, we put it in field for 1 year at winter, because you cannot drive core in summer here. Soil is like very, very, very hard. So we put it in for 1 year, and we retrieve it as whole after 10 months before next rain. And we also took soil, sieved soil, and we all irradiated by 25 kilograde twice. We have a radiation center here, so we can do this. And 25 kilograde should kill all microbes. And microbes be intact. It's not autoclave, they just killed, they're not alive. And we have naturally dry soil, which is the same treatment, everything's the same. And yeah. And so what we did first, we wanted to see if our soil is active. Nitrification potential is standard procedure. Using ammonia, you measure nitrate concentration increase. Irradiated soil, non-irradiated soil. Yellow is regular soil, whatever this color is, irradiated soil. And you see that irradiated soil have no production of nitrite over 20 hours. Nitrification potential usually used for snapshot of activity of microbes in soil. About 4 hours you should do this in real world. We did it for 20, and still you have no production of nitrate. Some production of nitrate in regular soil happened. It's weeks after irradiation. And then we took more than 6 months after radiation, like a few weeks ago, we just did N2O production potential. N2O production potential represents active denitrificants who are active in this soil. We just adding a lot of carbon, no oxygen. They start to do denitrification because it's anaerobic breathing, anaerobic respiration. And you can see that irradiated soil had a lot of N2O production in the beginning, within the first hour, and then stopped it. But this soil, regular soil, behave like regular soil should behave, exponentially increasing in activity. And this is N2O production in soil, ppb per jar. Some doesn't matter what does it mean, but what is interesting is dynamics. Dynamics here it looks like abiotic, and this looks like biotic dynamic for me. So we went now and we did rewetting with this course. It's a little bit longer. So the results I'm showing here, results of multiple experiments with multiple cores, and you need to take it into consideration that soil is very heterogeneous. You have some, because soil is heterogeneous. Okay, and each error bars is different cores. You cannot do experiment with multiple cores just because of logistic problems. And you see, this is our live soil, this is our dead soil, and we have emission of N2O in dead soil, and this dead soil is dead by radiation. So I hope they dead, at least, yeah. And when you're looking CO2, you don't see any difference. Means that, and we know this because it takes a very long time to start to be active, because soil is very dry, 1 year outside of soil, no water for a very long time. And CO2, if we will keep it, you see here we're keeping it 1600 minutes, mean it's many hours. And then we have some production in live soil, but no production of CO2 in dead soil. For me, it means that the dead soil, and live soil. But N2O producing in both doesn't matter what kind of soil you have. So you don't need to be alive to have emissions of N2O. And so we did also some experiment in jar, and we added water, and here N2O production picogram per gram per minute, N2O produced in control soil, alive soil, and irradiated soil also produced less, but produced because soil variable, so some soil producing more, some soil less, and CO2 other way around, so not produced in control soil, almost not produced in irradiated soil. And if this produced, I have some thinking that we have some abiotic production of CO2 because we have carbohydrate, a lot of carbonates, and we adding water, and we have some bubbling potentially. But again, this is, so we have 3 lines of evidences. I'm already writing paper in my head. So one is field, very fast. Second, jars, irradiated, non-irradiated. And third, cores, undisturbed cores, irradiated, non-irradiated. All of them show the same, very fast production of N2O after rewetting, and probably not connected to any microbes who are alive. Which is coming back well to this, because if we have an ammonia oxidizing to hydroxylamine, it is in periplast of bacteria because they're gram-negative, and when they're blowing up, you have a lot of enzymes released to the soil with water, and there they emit a lot of ammonia because you have ammonia, a lot of ammonia build up in soil in dry environment, and all this can happen. So, to conclude, a very short conclusion. Based on our research of past 2 years, I believe we have very strong evidence of abiotic origin of N2O production produced after rewetting of dry soils. Yeah, so this is what it is. Thank you. And I have time for one question. I was longer than. Yeah, I think what happens is following. We have gram-negative bacteria, have two layers of membrane inside of soil, and they're ready to work because ammonia oxidation happens very fast after rewetting, and it's pretty well known. It's not like only my finding. And once we have all these microbes who start to be leased by water because they cannot keep osmotic pressure, you have release of these membranes and parts of enzymes, and these enzymes, ammonia oxidizing enzymes, they're in periplast, they're floating there. They're released to the soil, and in this soil they emit ammonia, and they're producing hydroxylamine. Hydroxylamine very difficult to measure, and it's very active, but we have addition of hydroxylamine, and it's showing that hydroxylamine plays a very important role. once hydroxylamine is produced, it's meeting iron-free, which is rust, iron-free, oxidized iron, and start to produce N2O by reducing oxidized iron to reduced iron, which is hydroxylamine does chemically. And once it chemically does, we have oxygen in soil, and this iron oxidized back to iron-free. And this is my theory, at least, of mechanism, which is I will be very happy to show in lab, but I don't know how. Yeah. Oh, it's not agriculture, it's desert. No, they're not important for global radioactive budget. But they're small, but they kind of, they're small, but they're not very small. They kind of what? 300 nanogram per meter square per minute. So overall, these emissions are important for nitrogen cycle in deserts because these emissions covering about 30% of all total emissions in 1 hour, which is important. But is it important for global cycle of nitrogen? No. But it can happen in other soils. We just need to find it out. And more important for me is to explain this very fast emission of N2O after rewetting, which is like kind of puzzle. It's kind of a mystery because since Birch published in 1958, we're spending 60 years just to understand CO2, and we haven't started to understand nitrous oxide. And as nitrogen person, I do like to do this. Thank you. Now,
okay, so this is my proposal here, working with folks at BG Negev and Ilya's lab and collaborators, University of Tennessee. This is going to be a pretty short summary of the main results of this. Topic is unraveling the biogeochemical mechanisms of drought and rewetting induced nitric and nitrous oxide emissions from dryland agriculture. Similar to the research and the results that were just presented, but looking at growing wheat fields. Same field as was done with the earlier research and results. So drought and rewetting, why does this matter? Well, arid land systems, about 45% of the land area is projected to have an increase of periods of drought and changes in intensity of precipitation, either less precipitation over time or more extreme amounts of precipitation. So as you can see in this figure from 2016, large portions of the world's surface. Obviously we're here in Israel, but this would be the Sahel region, portions of the prairies, Australia, etc. So this, in looking at the impact of water balance on the nitrogen cycle is quite important. We don't have a mechanistic understanding of these processes. We don't know how to quantify the different rates and understand some of the mechanisms, either be it abiotic or biotic. Agriculture itself is the largest consumer of nitrogen fertilizer. So if we have an increase of losses of nitrogen, and we'll talk about the particular gases, but if we have an increase of loss, this could have impacts both economically and ecologically. So we really need to understand, as climate changes by the year 2100, with an increase in 1.5 to 2% degree temperature increase, what is the effect on nitrogen? So here is a figure that kind of illustrates the entire proposal and the research that I've done the last year. We're looking at, as I mentioned, drying and rewetting. So look at the soil carbon and nitrogen pools in both the effects on the plants and in the soil. So two gases of interest, nitrous oxide and nitric oxide. Nitrous oxide is a very potent greenhouse gas. Not a huge amount of concentration in the atmosphere, but is 300 times, it's called the global warming effect. It's 300 times more effective at trapping infrared radiation than carbon dioxide. Nitric oxide, on the other hand, has phytotoxic effects. This is another soil gas that's released with nitrogen pools into the atmosphere. This interacts in the troposphere to inhibit ozone formation, which can have impacts on human health and plant crop health. So these are the 2 soil gases of interest: greenhouse gas, phytotoxic kind of a gas. As you mentioned, there's a lot of research done on carbon dioxide and methane. We're in the desert, so methane isn't as applicable, but not as much research is done on nitrogens. That's why we're focusing on these 2 particular gases. The effect of water stress is also important. Obviously, if plants are stressed, they're going to have less exudates of carbohydrates. That's going to influence the pools of available carbon, which will have effects on metabolism of nitrogen as well. So looking at the drought climate feedback is important to see if we can get some mitigation strategies, and then also to understand the effect of drought. So going in once again on the microbial side of it, most of the results that I'm going to show today are just fluxes, or just the nitrogen fluxes, but collaborators in Tennessee are going to be looking at microbial, more of the microbial side of this. So this presentation doesn't talk about it, but I wanted to give a brief overview, especially for those who aren't as familiar with the nitrogen cycle. We're mainly looking here at nitrification and denitrification. As Ilya mentioned, in dry soils, there's large pools of ammonia that can be persist over time, can be maintained over time. Once you induce water, these pools can quickly, you can have quick nitrification and denitrification. Nitrification, this process of taking ammonia to nitrate, most often occurs in dry soils. This is going to be the largest driver of the NO fluxes. Whereas denitrification often occurs under anoxic, more mesic, so less prevalence of oxygen, more water, you get denitrification. That's a large pool for N2O. So different microbial processes govern different soil gas losses, and these processes are driven most often by available water. So we should see with pulses of water and the subtraction of water, these processes occurring differentially and as such different NO and N2O emissions. We need to quantify those processes. The other, so this study is looking at 2 particular nitrogen gases in agricultural systems. We've combined this, and I'll talk about the methodology in a second, but we combine this as well with soil texture. The first presentation kind of talked about this. This is an isohit of Israel. This is a rainfall distribution in Israel. Israel's a very small country, huge rainfall gradients. So this is just a figure from 2013 in the Journal of Climatic Change. And you can see us here on the bottom there, Stdt Boker, that's where we are. Huge differences in rainfall distribution, differences in soil texture. And these different soil textures may have different fluxes. And drought may impact that seasonality is important as well. Suffice to say, Israel is a really good, it's a good kind of laboratory. This is field studies, but Israel itself, you can see it as kind of a laboratory. You can take soils from different parts of Israel and be able to modify the texture and to see if we have different responses. That's what we have done. So, our experimental design, that's a brief overview of why we did this research, why it matters. The actual design we have 3 separate soil texture soils from 3 different regions among Israel. One in the northern portion of Israel, Magal, Rehovot is kind of more central portion of Israel, and then our campus here. Different rainfall gradient, different soil types. We have a sandy loam, a clay, and less soil. So 3 different soil textures, and we wanted to look at rainfall manipulation. So what we did is we installed these rainout shelters in half of the plots. We got 24 total plots. The way we did this is similar to the last presentation, and our lab does the same kind of standardized methodology. We have, that's interesting. Now it's frozen up on me. There we go. So we have these little these collars, these soil collars that have been placed into the soil. And this experiment ran for an entire year. We're able to get both N2O and NO data differentially with LGR, which is, it was talked about in the last presentation. But there's a flow-through system and there's an accumulation system. So two different gases. At 2 times of the year, we also induced a state of drought in about half the plots. So 12 plots had drought, 12 plots didn't. 2 times the year. This is right after nitrogen fertilization and at the flowering stage. So we want to see the effect of drought on nitrogen, but also the effect of nitrogen pools. Once you've added fertilizer, obviously the amount, and we did a urea fertilizer, so this is going to, once you have water, that's going to quickly change to ammonia. You have a larger pool of nitrogen. Seemingly the effect of larger nitrogen pools in the soil should have an effect on the penultimate gases, the penultimate flux. So temporal and spatial scale differences. Unfortunately, this graph didn't seem to go through. I ran this on a Mac, and this is on a Windows computer. But it's the way we establish these fluxes. You do a linear interpolation of it, and you can get a flux. That's for N2O. Okay, good. And then for NO, it's a little bit different. So that's very standard methodology of linear interpolation. To calculate the NO, because this is not an accumulation chamber, this is exhausted out to the air. It's the same thing. So this is one of the first data sets that we have. This is in December. And so you can see we have relatively large NO peaks. And the way we established these ones is we took the flow rate of the system and then the differences between the peaks, the maximum and the minimum. So that's how we derived the data. So what are our objectives? We want to determine these emission patterns. We believe that rewetting after in-season drought will cause greater emissions, whereas non-growing drought will cause less emissions. If you got more nitrogen in the soil, you have higher emissions than if the nitrogen has been released. So and this will have implications for industry, and we'll talk about that after I've shown you the results. But there should be some temporal dynamics. We want to understand the conditions dynamics so we can predict the effects of sustainability, economics as well, as I was mentioning. So what do we find? 2 main figures here. This is the soil type. This is 3 gases of interest, CO2, NO, and N2O. We found that texture had an impact. There was differences. But it does vary between the gases. So as you can see, some of them are not statistically significant, some of them are, most of them are statistically significant. So for CO2, for example, there was a difference in the total efflux of sand compared to clay, or less compared to clay, et cetera, et cetera. So texture had a difference, and yet the difference of texture played out differently for the different soil gases. Our collaborators at Tennessee are going to follow up on this with a microbial study, but we believe it's different microbial activity that is driving this. So nitrification, denitrification, respiration will have differences, different roles to play because of texture. But nonetheless, texture is important. Texture also has an impact on rainout. So for this presentation, I've only showed general rewetting. We also found there was a drop down in the second rewetting period. But for the most part, there is a difference between, for all the gases, for the rainouts and texture, and also, as would be expected, when you induce drought, the amount of efflux is reduced as well. So what do we find in conclusion? N2O and NO emissions differ significantly across textured soils. Each gas is affected differently. That's probably because there's different microbial processes. The drought response was also affected by soil texture. So texture is the chief controlling variable. The soils are exhibiting differences in these emissions despite identical climate and management. So texture is really important. Where soil processes affect biological processes, texture is important. And this is not really well documented as much in the literature. A lot of emphasis on microbial processes, not so much the soil itself. So why does this matter? Well, so if we have increased losses due to drying and rewetting cycles, there can be an asynchrony. So we saw a larger efflux at the beginning when the soil pools are highest. And this is just lost. When you have gases that are released, this is just lost amounts. You're growing a crop, you want to apply fertilizer to bring that into the plant to get increased yield. You don't want that to be lost to gases, both for an economic perspective and ecological perspective. This is a greenhouse gas. We don't want this to accumulate in the atmosphere. It's going to warm up the earth. So seeing these values, we should maybe affect fertilizer timing and when to apply fertilizers. And once again, this is going to vary based on texture. So if you are a farmer in the middle of Israel, your application of fertilizer will be different than if you're a farmer in southern Israel or northern Israel. Also, with better quantification, we're going to get another year's worth of data. We can also help with the models. Because N2O and NO flux in particular are relatively not defined. They haven't been studied as much. And so the modeling outcomes, the modeling numbers are quite skewed. So getting better quantified data will help with that as well. And yeah, that's the presentation.
So thank you, Ilya, for inviting me. Here's my first trip to Israel, and I'm having a fantastic time. Just wish I could stay longer. If there is any incoherence in my presentation, I'm going to blame it on my jet lag and not me personally. And the first thing I guess we should say is when I was putting this talk together, I think the content of the talk now doesn't exactly reflect the title. I want to be talking about drying and rewetting pulses, which is a common theme we've seen here today. And I'm going to be talking a lot more about carbon cycling. And I wanted to give you sort of a preview of where we're at in my lab with some highlights from 3 different experiments, only one in an arid ecosystem. The others are from Tennessee, but hope to convince you that the processes that we're looking at are the same regardless of the climate where we find these soils. So as has been mentioned here already, we are interested in how biogeochemical cycles respond to disturbances, both chronic and pulse related. So those can be, like I'm interested in the chronic change over time due to global warming and the stress that puts on environments. But also we know that's going to lead in most places to more intense, more frequent and intense droughts. So there's going to be changes in precipitation intensity and frequency in a lot of ecosystems. And my talk is about, or I study microbial dynamics, and so I wanted to tell you why microbial dynamics matter, although I feel that's been covered by quite a few talks already. But maybe a different perspective that hasn't been mentioned yet is that we need to think about the difference between access and allocation by microbes. So what I mean is microbes they need to have physical access to substrates in order to decompose them and to utilize them and either turn them into more microbial biomass or to convert to CO2. And then they have to decide what to do with that carbon. And they can allocate it to various things. They can allocate it back into growth. Some gets lost due to mortality and to metabolic functions and is lost as CO2. but some they can use to directly deal with stress, so they can secrete extracellular polymeric substances in order to change their hydraulic environment and tolerate, to some extent, drought stress. They can also choose to invest resources in the production of exoenzymes that allow them to go out and harvest more resources. Thus, they can allocate carbon to increase their access. There's a feedback effect there, and we're interested in how this microbial response can drive ecosystem processes, because these allocation decisions are going on at a micro scale over the entire landscape. And we're looking at the aggregate of all of those allocation decisions that microbes access. And I think it's been abundantly clear from all the talks we've had today that we don't really fully understand and haven't really fully quantified what those processes are, and they're in many cases not intuitive. So I'm going to add my own extra layer of complexity onto everything we've heard today already. But how does that access and allocation relate to precipitation frequency or intensity? So those access and allocation decisions are going on over this hydrological landscape, and this is just published from Parker and Schimmel paper more than 10 years ago, looking at what happens in a wet soil versus that same soil when it dries out. So when conditions are wet, soil pores are filled with water, microbes have access to all the substrates they need, but then conversely, their predators and pathogens have access to them, versus in a dry soil, many pores will dry out, but there'll still be some micropores filled with water, but also water films in larger pores, and it breaks up this hydrological landscape, and microbes can then become isolated from their substrates. They can also become isolated from predators and whatnot. And so drying and rewetting changes microbial access to substrates. It may also change how those substrates are transformed, the allocation decisions that microbes make. And those allocation decisions lead to either accumulation or stabilization of soil organic carbon. So that soil organic carbon can be either stabilized, potentially stabilized by those decisions. So we can have carbon compounds with a longer turnover rate, or we can have carbon compounds that have a shorter turnover rate. And that's some of the stuff that I want to show you. I have three highlights from 3 experiments to show you that kind of address these questions. So how do the dynamics of labile carbon and nitrogen, I wrote this when I had a lot of nitrogen data in there, but I cut that out for time. How do they respond to moisture pulses? Are there feedbacks with soil structure? So not the texture of the soil, but the aggregate structure of the soil. And then does that microbial response to moisture pulses, does that change the carbon, how carbon substrates are transformed in a soil? And do different substrates lead to different transformations? So we'll start with a semi-arid ecosystem. This is Mediterranean savanna. I did this work when I was a postdoc at UC Santa Barbara. So this is just outside of Santa Barbara, and we have a Mediterranean climate there, very similar to here. Wet winters, dry summers, so wet winters, dry summers. And sort of the highlight of that research was that the longer soils stay dry, the more CO2 is released upon rewetting. We've heard a little bit about the Birch effect. This is not necessarily a controversial finding. We find this in a lot of systems. So this is just the CO2 released upon rewetting, the total amount of CO2 released upon rewetting, and then the number of days that soil stayed dry. And then we see, at least for soils in the top 20 centimeters, we see a nice linear relationship where the amount of CO2 released increases with the amount of days that soil has stayed dry. And although that relationship does break down when you look at soils deeper in the profile. And another thing that we followed up on was looking at the physical structure and how the physical structure of that soil, that aggregate structure, might act to protect carbon substrates from decomposition, because carbon can be locked up in soil aggregates and be physically inaccessible to microbes. So this is just an experiment where, again, we looked at the respiration released from both crushed and intact aggregates with multiple cycles of wetting and drying. So from 1 to 5 cycles of drying, and then looking at the CO2 released, and we can see that with crushed, so these are intact, these are crushed. When you crush the aggregates, you destroy that physical structure, you get more CO2 released from microbial decomposition relative to an intact soil structure. But after 4 or 5 pulses of wetting and drying, you tend to either break down or slake those aggregates, you break down that structure, and you lose that protection from soil aggregates. So with increased precipitation frequency, we may see less stabilization of soil organic carbon. And these are the things that I followed up on when I moved to University of Tennessee and continued my research. Tennessee is not an arid or sub-arid ecosystem or climate by any means. It's a sub-humid, mostly deciduous forest, converted now to farmland. We get almost a meter of precipitation, evenly distributed throughout the year. But again, I was interested in the moisture pulse dynamics in these soils because it only takes about 3 to 5 days of no rain for these soils to get as dry on the surface as those dry soils we saw in Southern California. And we also looked at the effect of conservation management, so changing fertilization rates, using winter cover crops, either nitrogen fixing or non-nitrogen fixing cover crops, and reduced or zero tillage. And then looking at this same biogeochemical cycling. So this first experiment I want to tell you about was done by Aubrey Fine, who's a PhD student in my lab. And what she did was she took 3 different aggregate fractions, large macroaggregates, small macroaggregates, and microaggregates. So these aggregates go from about 2 millimeters down to 250 microns, and then microaggregates smaller than that. And then she incubated these aggregate fractions by rewetting, and then rewetted them to about 50% water holding capacity at varying frequencies over 42 days. So we have all of these different pulse frequencies. We have a control with 0 water added, only 1 rewetting pulse over 42 days, 2 pulses, 4, 8, and then this 32 pulse, which really represents a constant wet situation because the soil never fully dries out from the previous pulse. And then you can see this destruction, at least in the large macroaggregates, you do get that destruction of the physical structure over time. And we looked at the same respiration that occurred with rewetting, although we did it a slightly different way. We looked at the ratio of that CO2 released in the final pulse compared to the initial respiration rate. And you can see that with the number of rewetting events, that ratio decreases. This is just a different way of showing that same data set we showed from California, in that the longer the soil stays dry, the more CO2 is released upon rewetting. It's just a different way of expressing it. And then we did find that Soil structure did matter. Large macroaggregates did release proportionally less carbon dioxide relative to microaggregates, although the difference isn't especially large. And we measured about a dozen different biogeochemical variables. I'm not going to show them all one by one. This is just an example. This is extractable organic carbon in potassium sulfate, and then permanganate oxidizable carbon. These represent two relatively labile, easily decomposable pools of carbon in the soil for 3 different aggregate fractions. And you can see that pulse dynamics, sometimes they matter, sometimes they do matter, sometimes they don't matter. We found varying patterns with different variables, and it gets very hard to analyze coherently without reverting to some kind of multivariate analysis. So this is the full range of all the variables that we measured, from carbon and nitrogen pools in the soil to extracellular enzyme activities and even phosphorus. And we can see that they're spread relatively evenly over this multivariate space. And then when we look at the actual data points, they're colored by pulse frequency, and then the shapes, circles, diamonds, triangles, represent the different aggregate fractions. It's a little difficult to see these patterns, but what it boils down to is that we see that aggregate size sort of sorts along this axis, rewetting frequency along a separate axis of 90 degrees in this multivariate space, indicating some different controls on biogeochemical variables. So if you've got, say, large aggregates and a high rewetting frequency, you're going to be in this corner of the plot, and you're going to be more sensitive to peroxide oxidizable carbon, nitrate, and nitrification. But what it boils down to is we found that rewetting increases CO2 and nitrogen mineralization, and it does stimulate the production of relatively labile forms of carbon. Aggregate size seems to scale with extracellular enzyme carbon and nitrogen stoichiometry. So the enzymes responsible for carbon mobilization versus nitrogen mobilization, the ratios of those activities tend to change most with aggregate size. So what we think is happening here is we get destabilization of soil organic matter with little aggregate protection. We need those aggregates to protect that soil organic matter in the face of increasing moisture pulse frequencies. And aggregate size seems to be related to increasing microbial nitrogen and phosphorus limitation. We're not exactly sure of the exact mechanism there, but it's definitely something that we're going to be following up on. The third and last experiment that I want to talk about, we asked the question, do the carbon substrates matter when we look at adding a carbon substrate and having that substrate be transformed by microbes into different pools of carbon, either relatively labile, soluble molecules or microbial biomass versus things with a longer turnover rate like microbial residues, mineral associated organic matter, and aggregate occluded organic matter. And we asked the question, well, if we add different types of carbon substrates, subject them to different drying, wetting frequencies, and look at the different conservation management practices, might we see a difference in this carbon allocation? And so we did this again in West Tennessee, utilizing a long-term experiment that's now been running for actually more than 40 years, that has a full factorial design, including 4 different cover crop treatments, 1 non-nitrogen fixing winter cover crop, 2 nitrogen fixing cover crops, no cover crop, and tilled and no-tilled treatments, and then 4 levels of nitrogen addition. Although for this experiment, we just focused on tillage and 3 of the cover crops. Otherwise, we would be looking at a huge number of plots. We cut our plots more than in half because 128 is just too much to look at. And this was another PhD student of mine who did this experiment, and this one has been published, so we can actually you can actually look it up if you want. And she took 6 different conservation management treatments, 4 drying and rewetting cycles with 3 replicates, and then that gave 72 jars. And for 72 jars, she added labeled glucose, another 72 jars, 13C labeled corn residue, and then incubated them for 28 days, and then looked at where that stable isotope label found its way into different carbon pools in the soil. And so I'm just presenting you with sort of the final results from her paper. Oh, sorry, the reference got cut off at the bottom here. But what we found was for a large number of biogeochemical variables, some of them did vary with moisture pulse, so the bars are different moisture pulse frequencies. The different sets of bars here, each one of these is a different conservation management practice. And the practical upshot was that moisture pulse seemed to be much more important for controlling carbon substrate utilization than management practice. And the pattern's the same for each one of these management practices. They only vary by a little bit. So the other thing that she did was she used structural equation modeling, another multivariate analysis, to try to look at the relationship of each one of these drivers to each one of these biogeochemical variables. And so this graph is just for adding glucose-derived carbon to a soil. And you can see that moisture pulse can either have a positive correlation, so that's the black arrows, or a negative correlation, which is the gray arrows, with these other variables or other treatments. And then you can see the sort of the directional relationship, whether the relationship is a direct one, say between moisture pulse and extractable organic carbon, or is it an indirect one, like between moisture pulse through microbial biomass carbon to bulk soil organic carbon. And what we found was for glucose, a very complicated structural equation model. I don't want to go through all the arrows, but the main things we found was that, yeah, soil structure was actually directly influenced by moisture pulse. The greater the pulse frequency, the more carbon was stored in the microaggregate fraction. The greater the moisture pulse frequency, the less carbon we found in extractable organic carbon. That's actually this figure here. But if we take a slightly more complex substrate like corn-derived residue, we get a much different relationship. We're still adding carbon, we're still subjecting it to the same moisture pulse treatment, but we see some distinct differences in the effect of moisture pulses on the carbon allocation in that soil. So instead of a positive relationship, which we saw with glucose, we now see a negative relationship with microaggregate carbon, but we see the same negative relationship with extractable organic carbon, and then a much different relationship between extractable organic carbon and microbial biomass than compared to, say, a glucose treatment. So I don't want to get too hung up on each one of these arrows. Just suffice to say that the pattern is very different. The significant variables are very different between these two carbon substrates. So again, in my jet lagginess, I did forget to add a summary slide. So just in summary, I will say that aggregate structure seems to be a key protective or a key stabilization mechanism for soil organic carbon in the face of increasing moisture pulses. And we also find that that soil structure is, again, very sensitive to moisture pulse frequency. And that we need to think very strongly about when we do these labeling experiments, what kind of carbon substrates are we using? Most studies use glucose to trace carbon through into soil organic matter. And that may not give us the full story of whether or not carbon is being stabilized in the face of different moisture pulse frequencies. So I will leave it at that. Thank you very much.
And what when you add the carbon, form carbon source, in which form did you add it relatively to glucose?
We added the same amount of carbon. So it was ground corn litter, but it was roots, stems, and leaves. So roughly equal proportions of all three of those. We got it from a colleague. It was ground to pass through a 1 millimeter sieve.
Your pulse frequency, what you call pulse frequency, sometimes you call it pulse number or number of pulses. So is it frequency or is it number of pulses? When you say something depends on the number of pulses, meaning the more pulses you do, then the bigger that thing gets. So or is it the frequency?
So for both, yeah, thank you for asking. It would be different. So pulse number could be the same frequency, just multiple number of pulses. For those last two experiments, the labeled substrate addition and the aggregate fraction, those were both done, all the incubations were done in the same amount of time with different numbers of pulses. So the frequency is what we're changing. The more pulses, the shorter the frequency. Whereas for that very first experiment with the aggregates in California, that was just pulse number. Good catch.
So maybe it's very much about the methodology, but are you separating the microaggregates from macroaggregates? I'm wondering if macro aggregates don't have a different composition of carbohydrates which keep them aggregated as compared to microaggregates which don't have so much sticky carbohydrates that stick them together.
That's what we think may be a possible reason. There may be very different carbon compounds found in macroaggregates versus microaggregates.
The compounds, not the bacterial. Yeah.
That could be different too, but remember that macroaggregates are composed of microaggregates that are glued together. So you might think of it as microaggregates might be, in terms of a microbial community, they might be a subset of the macroaggregate community. So a macroaggregate community actually probably has more potential niches for microbes to inhabit.
So following up on David's question, so the aggregates are soil dependent. So if you had different soil types, how would, and you would assume that the microbial community would change, how would that affect these?
So the question is, if I had different soil types, how would I expect the microbial community? Huh. I would expect, at least for these the aggregate dynamics, I would expect that they wouldn't change as much as if we were to look at soils from, say, different climates or soils with very different textures, say a sandy soil versus a clay soil. But I think we're finding in the more ecosystems we look at these dynamics, there are more commonalities than differences, although it's very hard to say that because everybody is doing slightly different types of experiments with different parameters, calling pulse number, pulse frequency, and it can be very hard, I think, to tease that apart. But what I hope that we come down to is maybe trying to find some generalized principles that apply to all systems. I don't know if we're there yet.
In your first part, when you look at activity of the size fraction. Do you recognize, do you see any specific enzymatic activity related with different grain size?
Again, we didn't look at grain size, so I can't tell you, I can't, we would looked at microaggregate size, which is different from structure. But we did see different proportions of enzyme activities in different aggregate fractions.
If there are any associated that you recall that phosphorus is bigger or whatever.
It looked like we saw increases in nitrogen and phosphorus acquiring enzymes in macroaggregates, which would suggest that maybe there's a limitation to microbial growth.
So good afternoon, everybody. My name is Shahar. Thank you for staying here, whoever is here. So, as a colleague of Ilya who is working on gaseous emissions from soil, the question I ask myself is, what do I actually measure relative to what is actually going on there? And that's what I'll try and somehow show you. The work was done with collaboration with other researchers from the Volcani Institute, where I work, which is Dadi Rusu, Shmulik Friedman, and Shahar Baram, and Alon Gal was part of his master's thesis. Which slide? So, in the general frame, we were talked about today, but everybody, I assume, have seen these graphs. Basically, global trend of greenhouse gases. We're not doing so good. That's an obvious fact that everybody knows. And when we look at that, I ask, who contributes? How do we quantify the contribution? And can we mitigate it? And in order to be able to answer the last one, we need to first to be able to answer the first two. And eventually, as scientists, we would come to the legislator, whoever, and they would come up with these big pie graphs or whatever that would try to quantify the contribution from every sector, saying who is putting what. And this is just the nitrous oxide in the US from 2017. And you can see nitrous oxide is mainly due to agricultural activity based on what it says here. So how do we go about measuring it in the field? If we want to go and measure fluxes, we have the two common methods. One is the eddy covariance tower, which nobody spoke about it today, but basically it has a very big fetch, a very big area. It needs to be a very uniform area, and it's a very accurate method. It's a very good method to represent large scales. And then we have what we call the static chambers, or like we've saw now, that are very, very localized, which are good to look into different treatments, as we've previously seen today. And I would focus my talk mainly on these, on the automatic chambers and the limitations that we came across with them. So again, you've seen that. Eventually what we get is we get a temporal dynamic when we measure. And once we have these temporal dynamics, we would try to upscale it into the regional scale and so forth, and see what treatment would do best for mitigating what. The first challenge we go about when we try to set up a measuring system is we're budget limited. So we have to say, okay, how many chambers or stations can we put? And how do we account for spatial variability, like Sin just showed? So many different parameters show different variability in space. So how do we account for it? I don't have the right answer for it, but I'm just raising that question. So where should we measure? And this is just one example from there's so many different papers that show it. You can see that the potassium, the nitrogen, the EC, the phosphorus, the organic carbon, the soil pH, everything shows a different pattern, and this is just in the topsoil. So the microbial activity would be very different in each one of these parts. So what are we actually measuring at the end of the day? What does it represent? It's a question. So with that, we can ask ourselves, did we manage to capture the spatial variability, or did we account for it to start with? But this is part of the limitations of, we're limited by money, by manpower, by equipment, and all these. So normally what we do is we use the static chamber method. More advanced group today use devices like the Los Gatos, like what Ilya just shows, that basically what it does, you have the base that variable names that you call it. We call it the anchor, the base, the color. It doesn't matter. You take some sort of a ring or a square or whatever, and you place it into the ground. Basically, you have a defined area that you continuously measure from the same place. Okay, normally you would try to put it a few weeks, if you can, before you start, to let the soil stabilize, and then you start measuring. And then whenever you measure, you come, you cover it, and you circulate the air. If you have, you can do the analysis in the field. If not, you would come and you would put the cup, and you would take a syringe. This has been done for years. Commonly, this is the most common method. Okay? The reason these anchors were derived for originally was because of these methods. Because you don't know what the gas is inside. You don't know the gradient. So you have to close it for a reasonable amount of time so you'd have a reasonable a big enough buildup in the chamber that you can analyze. And then you have to prevent back diffusion. So these anchors were created. This was the origin of them, but still we use them, even when we use these sophisticated machines. And my question, what I would like to come and raise is why? And is it smart to still use these? And I'll try to convince you that I think we should stop, but we can argue about that. That's part of the fun. So our objective is to use, and especially in drip irrigation. And here we mainly work in drip irrigation in agriculture, so one of the questions is, what do we do in drip irrigation? So we did both field measurements and numerical modeling, so we have nice graphs, and to see what is actually the relationship between. So we look at drip irrigation. We have 4 scenarios. If we use an anchor, we can put the dripper inside the anchor. We can put it adjacent. Some works in the literature basically took the emitter, said, let's say it's a 4 liters per hour. So they put 2 liters per hour here and then 2 liters per hour here. So they split it. And in reality, this is what we have. There is no limitation. There is no anchor that limits any of the movement. And why does it, what's the problem here? Think about it that the emitter is here. So basically, once the water will go around, it hits the wall. It cannot keep on diffused to the side, so it would go down. And if the water is here, and we know that most of the biological activity happens in the top 5, 10 centimeters. And if you put the anchor here, water cannot penetrate. So we have 2 opposing mechanisms. So what is the actual effect? So we looked at it, you can see, in over 2 years in an orchard, part of a bigger experiment. We also checked that just to see what we measure. So what we did is we did adjacent, and we did a dripper inside, and we used the portable FDR in the field to measure continuously, and this is what it looked like. You can see this is the dripper inside, and this is the dripper adjacent, and this we didn't measure. This is the question mark. What is going on here? But we tried to assume what is going on there. And just looking at this is fairly simple. You can see here the ratio between basically what we measured inside, these are the 2 methods, and adjacent. And you can see a very, very, very big difference between the 2 methods. They don't give the same results. It's not surprising. There's not the same amount of water. There's not the same nutrients inside. But these are, and then when we try to extrapolate it and say, okay, what is the emission? Depends where we measured. And this just, now we go to simulations because it's easier when we do simulations. What I want you to focus is, this is times from when we emitted, and this is depth, okay? Each one is a different depth under the anchor, okay? And this is an anchor, I think, 20 centimeter anchor that we simulated. And what I want you to see here is, look, when we irrigate, and the hollow one, so the hollow shapes, is basically no anchor. The full ones, there is. You can see that there's a huge difference. Just looking at the water field pore space, there's a difference in what is going on within that domain. And this is integrating the top 10 centimeters inside the anchor, what is going on there. Okay? And if we look at it more visually, I think it's very easy to see. You can see these 2 emitters. This is a heterogeneous domain, a 2D domain, with a let's say, simulating the heterogeneity of the soil. And you can see very simple, these are drippers with no bases or anchors around them, and this is where it is. And you can see what happens when you look at the nitrate, you can see what happens when you look at the ammonium, and you can see what happens when you look at the water, and eventually what happens when you look, yep, yeah, sure, sorry. And you can see what happens when you look at the flux. So there is a big impact. And again, if you look at just the ammonium here, and you can see this is when you have the chamber, and this is, again, down the soil profile, you can see a very big difference here. The ammonium is always higher inside the chamber, while here you can see that the nitrate has very big fluctuations because nitrate is more mobile, so it moves with the water. So if you were just irrigating and not fertigating, you would flush it down and the nitrate concentrations would be lower and so forth. So another thing we said is, okay, what is the chamber size? Normally what people use is our small chambers because they're easily to construct. Normally you take off-the-shelf product, a hose or something, a pipe, you cut it, you have many, many chambers. But does the size matter? It does matter. It have a big impact. Basically, the bigger the chamber, the closer it would be to reality. But it's hard to operate big chambers, especially if you want many of them. So again, how do you account for the error associated with the size of the chamber? And you can see again, this is nitrate, this is ammonium, very big differences. And this is the size if we look at the NTO, and you can see the black line is basically what we assume to be the real measurements, and these are what we measure based on different chamber sizes. Okay, you can see a 20 centimeter, 40 centimeter, and 40 centimeter chamber. So very simply, if we put the dripper inside, during fertigation, it may increase the NTO flux by up to 50% almost, just by placing the dripper inside. And following irrigation, it may decrease the flux by almost 100%. It's just by where we place the chamber relative to the dripper. So what we tried to do is we tried to come up with a, I would say simple, some might disagree, kind of normalized reference table that would account for all the soils together. Basically what it shows is very simple. We take a unit that is called a capillary length, which is basically dimensionless. It can normalize all soils together. Basically a sand would have a high capillary length, okay? And text will have by large capillary length, basically it's 1 over alpha. So if you have a very small alpha, it would be a large number. And then you can normalize it and you can say what is the size of the chamber you like, and especially in drip irrigation, basically eventually you would say, okay, if we have two emitters that are close to one another, eventually their wetting bulbs would meet. So that creates like a wall anyway. So if your chamber size is the size of that wetting bulb, you're close to being in a good place. Okay? Because then you would have the minimal impact, but of course if you look at it from the 3D space, emitters are bound by 2 dimensions, only that length, while a chamber is radial, or both dimensions. So we'd always have an interference of some sort. There's no way you can go about it. So what I would like you to remember is very simple, that the true N2O is probably bigger than what we measure inside, and that's probably bigger, always bigger than what we measure adjacent. And eventually what we can do today is we don't need to use anchors. If you use chambers, and if you are not chambers, if you're using field methods that have very, very sensitive equipment, like Ilya just said, at the sub-ppb level, there's no need to use the anchors because there's so much noise that they put into the system that it's better to avoid it, and then you just randomly place them in the field. All you need to do is put some sort of a flexible system at the bottom. Anyway, they have a venting tubes, venting holes in them, so it doesn't have to be 100% sealed anyway. So just, let's stop using anchors. That's all I wanted to say. No. But that's what I'm working on now. We will, that's the next step. But I can say, just from what you asked, about a month ago, I had a meeting with, what's his name, Butterba, Klaus. And he said that they stopped. That they just take the chamber and they just walk around with it. And they just said they're using big chambers now. They just use WAG big chamber and they just walk around with it. Just because he said they figured out that that's the best way. Whether he has the data to prove it or not, I don't know.
Yeah, you can, actually I can open for discussion because we don't have any more speakers and we have 15 minutes time. So everyone who wants to say anything is welcome.
So, but it's more significant like in drip irrigation setting. Like if you do it here outside, if you assume it's more.
It's a question. The answer is in rain it would be more uniform, obviously, but you're limited by roots being able to go in. Many times, you would cut the plants because you cannot. So always when you use an anchor, you would have a disturbed surface. Always. Just by hammering it in. Okay, and then you can have preferential flow on the edges of it. There's so many, it just adds so much noise to a system that is already so noisy to start with. So why add that noise? Even automatic chambers, they don't need the anchor. Just put some sort of, I don't know. I mean, what I'm working on now, our experiment is we took a memory foam, whatever you call that foam. So we have a 6 centimeters memory foam, and we just place that. So it seals good enough. Not that we've seen yet, but we're still checking it. I mean, again, even if we have absorption, relative to what we have, the error associated with just the method we're using now, so it's kind of there is no perfect method. There is, I think, I mean, obviously, and you'd see still there'll be many studies coming from mainly the poorer countries that they would still use the old, I'm saying they would still use the old systems because from labor-wise, it's very easy. You just put many of them in the field, you go with syringe, you take samples, it's easy. What does it actually represent? That's a question open for discussion. I don't have an answer for it.